So where we left off in the last video was the structure of a ribosome. So remember, it's made of two subunits, the small and the large subunit, and it has three sites, the E site, the P site, and the A site. So we're going to talk about how those three sites actually put together the growing polypeptide chain. So there's three steps of translation, just like there were of transcription, same three: initiation, elongation, and termination. So we need to start the process somehow, we need to keep growing the polypeptide, and then we need to finish the process. So all of those three are the three stages. And each of them requires the help of proteins to get it going. So let's talk about initiation. So first, to build the growing polypeptide chain, we have to bring together the mRNA, which has the message in it, the both subunits of the ribosome, because they're separated, they're not together unless they're building a polypeptide, and the tRNA, a special tRNA called the initiator tRNA, which is going to be the first tRNA in our which is going to carry the first amino acid in our polypeptide chain. So what happens is we have our mRNA, and a small ribosomal subunit comes in and attaches to that mRNA. And the mRNA goes in 5' end first, and the protein gets, or the mRNA gets read in the 5' to 3' direction. So the 5' end moves this way first. And the small ribosomal subunit is reading this mRNA strand until it gets to the start codon. Now, every single polypeptide of every single living thing that has ever lived on Earth started with the codon AUG. That's how we start all proteins. So right here is the start codon. So the small ribosomal subunit read the amino acid until it got to the start codon, our AUG, and then our initiator tRNA, so that's the tRNA that carries methionine on it, the anticodon UAC, binds to the start codon, and then we have initiated we've brought the first tRNA in to start building our polypeptide chain. Sorry, I don't know what that's. Okay, so we're almost finished now. We have our small ribosomal subunit, our mRNA, and our tRNA, but we're missing the large subunit of the ribosome. So these special proteins, remember I said that there's going to be proteins that we need at every step along the way to help us. A special protein is going to go find the large ribosomal subunit and bring it in. And then it's going to fit the initiator tRNA right into the P site, and then it's going to keep reading the mRNA from 5' to 3' until it's done. So this right here is called the translation initiation complex, which means that that's the large and the small subunit of the ribosome, the mRNA, and the initiator tRNA, which carries the methionine. It matches up with the start codon. There's also energy needed. That's what this is. GTP is something similar to ATP. And so when it loses its phosphate group, it provides the energy through a coupled reaction for all of this to happen. You don't need to know much more about that other than that it requires energy, and it doesn't use ATP, it uses GTP. So we finished initiation. I'll talk about elongation. So now we need to add our amino acids to the growing polypeptide chain. We started it. We're good to go. We just need to now add our amino acids. So basically, we're going to add on amino acids one by one, and it's just going to keep growing, our polypeptide chain. Don't worry about N terminus and C terminus. Okay, so each addition involves proteins called elongation factors. So remember, we're using those special proteins again, and there's three steps: codon recognition, peptide bond formation, and translocation. So what happens is, here's our tRNA that's in the P site, and here's our growing polypeptide chain. Okay, so we have 4 amino acids on it so far, and our mRNA is going to move through as the 5' end is leaving and the 3' end is, or the 5' end is enter. The 5' end is on this end, and it's going to move along this way. So the next amino acid, or the next tRNA comes in. It matches anticodon to codon in the A site. Then it fits right into the A site, and the tRNA that's in the P site has the amino acid chain on it, and there's a peptide bond formed between this amino acid chain and this amino acid. And so what happens is now this tRNA that's in the A site now has the amino acid chain on it, and this tRNA is done. So they move over one spot. This tRNA now moves to the P site. It's now in the P site and now has the entire amino acid chain on it. And this tRNA is done, doesn't have an amino acid anymore. It leaves to go find one of those proteins to add another amino acid back onto it and recycle. So the tRNA comes into the A site. The amino acid that's attached gets a peptide bond formed between the amino acid chain that's already there. Then they both move over. So the tRNA that was in the P site goes to the E site and leaves. And the new tRNA that's in the A site now moves to the P site, and it has a growing amino acid chain attached to it. And you can see here that there's also some energy required in these steps, again using GTP instead of ATP. Did I miss anything else? And translation proceeds along the 5' end to the 3' end. So basically the 5' end goes into the ribosome and moves along. So it's just like how we read left to right. It reads 5' to 3'. All right, so we're done with elongation. Let's talk about termination. So we're building this polypeptide, this protein subunit, and we need it to stop eventually, right? It only is a certain amount of amino acids long. So what we're going to do is we're eventually going to need to tell the ribosome to stop building a protein. And what happens is the ribosome is reading the RNA, and as soon as it reads a stop codon in the A site of the ribosome, it knows that it needs to be done. So, do you have a picture? Okay. So basically, here's our growing polypeptide chain. Here's a tRNA that's in the P site. As soon as this A site reads a UAG or a UAA or a UGA, it says, okay, polypeptide's done. We need to get this thing out of here. And what it does is it accepts a protein called a release factor. So it doesn't take a tRNA. Instead, it takes a release factor. This, again, is one of those proteins that we need to help us do all these jobs. Remember, we talked about the proteins that bring in the large subunit for initiation. We talked about the proteins that help bring in the tRNAs for elongation. And now these proteins, the release factors, help us with termination. And so basically what happens is this release factor comes in, and what it does is it brings in a water molecule because we need to break the bond right here. So this amino acid is bonded to this tRNA. We need to break that bond. That way this tRNA can go on its merry way and our polypeptide can go do what it needs to do. So do you remember so far back, when we want to break a bond, we put in a water molecule through hydrolysis and the bond is broken. So this release factor brings in a water molecule to sever the bond between this tRNA and this amino acid. And so that's what happens. The bond is broken and then this polypeptide is done. And then everything disassembles. So the mRNA is done. It's going to go someplace else. The release factor leaves, and the two ribosomal subunits leave as well. So everything is done due to that release factor. Okay, so everything we've been talking about so far is one mRNA and one ribosome. But what actually happens is there can be many ribosomes reading one strand of RNA at the same time. So if we start reading one strand of RNA, say this is a strand of RNA, this is the ribosome, my fingers are ribosome, it starts reading it, 5' end, 3' end, starts reading it. Once it gets to right here, there's nothing stopping another ribosome from binding right here, so another one's going to bind and they're both going to move down the chain together. And as soon as it's free again, then another one's going to come back here and bind. So a bunch of proteins can read the same mRNA strand and build the same polypeptide at the same time. This helps make a lot of gene product in a short amount of time. That's how your body can so quickly make proteins, because of this multiple ribosome mRNA complex called polyribosomes. So poly means many, ribosomes are ribosomes. So polyribosomes are many ribosomes attached to the same mRNA making the same polypeptide simultaneously. So here's our mRNA, this red strand right here. This is the 5' end. That's where the ribosomes bind until the 3' end. So basically this was the first ribosome. It started right here. It moved all the way down, and now its polypeptide's done. This one started a little bit afterwards, so it's almost there. This one third, this one fourth, and this one, as soon as there's space, these two ribosomal subunits are going to go through the whole initiation process, and they're going to join right here and move down the strand. So you can see that right now this mRNA is being made into 4 different polypeptides. So it's working 4 times as fast as if only one ribosome could bind at a time. So that's really key. That's how we can make proteins so fast. So here's an actual picture of it. You can see that you can kind of see the skinny strand moving through this, and that is the mRNA, and then these are the ribosomes. All these dots are ribosomes working on that mRNA. So this happens all the time. Okay, so when we're done after termination, we have a free polypeptide. But remember that polypeptides aren't necessarily functional proteins. So there might be some things that happen to the polypeptide chain before it becomes a functional protein. They're modified afterwards, and they have to fold up. Remember that polypeptides are big, long strings of amino acids, but that's the primary structure, the polypeptide, but they have secondary, tertiary, and often quaternary structure. So all this happens after termination. So as it's being synthesized, as it's being built, the protein starts to fold. So as soon as it's coming out of the ribosome, so as soon as it's starting to build out of the ribosome like this, see how it's starting to fold? The R groups of the amino acid are starting to interact with each other, and they start to combine, right? So that's what happens to form secondary and tertiary structure. It starts to fold as it comes out of the polypeptide. Out of the ribosome. When it's done, it'll probably do a lot more folding, but it starts to fold as it's being built into its 3-dimensional shape. And then it may also have some modifications made to the polypeptide chain before it can do its job. For example, amino acids may be chemically modified, so let's add a carbohydrate to this amino acid, let's add a lipid to this amino acid. That way it can fold up to better do its job, because it needs to be a very specific structure to do its job. And some amino acids may be added or taken away at the ends. And they may also cut the whole polypeptide. For example, insulin is made from one polypeptide chain, but what happens is there's a middle portion that gets cut out, and now we have 2 polypeptide chains, but those 2 polypeptide chains, because we cut it in half and took out a little piece, the 2 polypeptide chains then join together, and the insulin has quaternary structure. So all of these sorts of modifications can happen after translation, and sometimes the polypeptides aren't ready to do their job until they get cleaved, until they get modified. So they get activated after termination of translation. And then this is when we get quaternary structure. So one polypeptide from one gene needs to find another polypeptide from another gene to fit together to make quaternary structure. And, okay, think back. We've been talking about ribosomes this whole time. There are free ribosomes and bound ribosomes. Now, we talk about them like that. The free ribosomes are in the cytosol, and the bound ribosomes are attached to the ER, but they're not stuck there. Because remember how they disassociate, and then they find each other again each time they have a new mRNA? So what happens is they're always free. So the mRNA is free in the cytosol, and they start the initiation complex. Now, proteins that are built in the cytosol are going to be used in the cytosol. They're going to be free. Proteins that are built by a bound ribosome are either going to be secreted or used in the endomembrane system. So the ribosomes are identical, and they can switch between free and bound. And so once we start building the protein, it always happens in the cytosol, always. Once the protein is a little bit built, the polypeptide chain, there may be a signal on it that says, okay, this needs to be bound. So let's go to the ER and continue our translation. So the synthesis will always finish in the cytosol, we'll always finish building it in the cytoplasm unless there's a signal, it's called a signal peptide, that says go to the ER so we can finish there. And basically what happens is, so this guy is being built in the cytosol, and here's our signal peptide. Signal peptide says, take me to the ER. So it binds to a protein called the signal recognition protein, right here, SRP. So this is the signal peptide. The signal recognition protein recognizes that. And then it says, okay, let's go to the ER, and it takes it to the ER. Then the ribosome binds to the ER, and the protein is made, actually, it snakes inside the ER. So the protein will either finish completely inside the ER, probably if it's going to be secreted, that way it can leave through a vesicle, or it might finish half inside, half outside the ER as a transmembrane protein. And then our protein is done.